Good day, BSMLS2 students of San Pedro College. This is Dr. Salaver, and I am assigned to discuss about enzymes. We will have two series of lectures for enzyme. We will have the first one this week, then we will have the second one next week. Let us first define what are enzymes. Enzymes are biologic catalysts that accelerate or speed up chemical reaction. It's very important to refer to enzymes as biologic catalysts because there are inorganic catalysts that we can use in the laboratory to speed up chemical reaction. But when we talk about enzymes, these are proteins present inside our body that speed up chemical reactions to keep us alive. That's why we need to refer to them as biologic catalyst. Now, how do enzymes speed up chemical reactions? They lower the activation energy, and we will discuss that later on. One unique characteristic of enzyme that I want you all to take note is that the enzyme is not consumed in the reaction that it is catalyzing. For you to understand that, please refer to this enzymatic reaction. We have here a dark blue colored substrate, and this substrate is supposed to be acted upon by this light blue colored enzyme. The substrate will form a complex with the enzyme, and after that, the substrate will be converted into the green colored and orange colored products. What do you notice? It is only the substrate that is chemically modified to form the products, while the enzyme is not consumed during the chemical reaction. Chemical reactions are comparable to a person carrying a huge rock up a hill and allowing that rock to roll down the hill and break down into pieces. The huge rock here is acting as the substrate, while the tiny pieces of rocks are the products. Now, what do we mean by activation energy? It is similar to the effort, the force that the person has to exert to carry the huge rock up to the top of the hill. So when you say chemical reactions, the chemical reactions will only start if they are provided with the force, with the effort, with the energy. And that's what I'm referring to as the activation energy. Activation energy is the energy needed for the chemical reaction to proceed to form the products. Enzymes speed up chemical reactions by lowering activation energy. This is your substrate, and these are your products. For the substrate to form the products, the substrate must be provided with an activation energy for the chemical reaction to start. In the case of a chemical reaction that is not catalyzed by the enzyme, the activation energy is very high. So, it would be difficult to start the chemical reaction. But, with the presence of an enzyme, the activation energy is lowered. That's why it would be easy for the substrate to undergo the chemical reaction and eventually become the products. All chemical reactions in the biological systems, so meaning inside our body, have an energy barrier which prevents reactions from proceeding in spontaneous manner. So the energy that we have to put into the reactants, into the chemical reaction, to start it is called the activation energy. And with the presence of enzymes, the needed input of energy will be lowered. That's why it would be easy for the chemical reaction to proceed. What are now the characteristics of enzymes? Enzymes speed up chemical reactions by lowering the activation energy. And even if you will add the enzyme into the chemical reaction, it will not have any effect on the products. So what do I mean by that? So take a look at the examples I placed in the PowerPoint slide. You have here a chemical reaction which involves substrates A and B and the products C and D. C and D are the products of the chemical reaction of A and B, and that's without the presence of the enzyme. Even if I will catalyze the reaction using an enzyme, the products will still be C and D. So therefore, the presence of enzyme will not affect the nature and property of the products. Unlike the catalyst that we are using in the laboratory, enzymes are highly specific in their action. So meaning they will specifically act on a certain substrate. So let's say for example, glucose oxidase will only act with the substrate glucose and will not act in the presence of galactose and fructose. Next, since enzymes are proteins and proteins are prone to being denatured, so expect that enzymes are sensitive to pH and temperature because these are two factors that can denature proteins. Later on, I will also explain to you guys how the substrate concentration can also affect the rate of enzymatic reaction. Enzymes are proteins, so expect the properties of proteins are applicable to enzymes. You know very well that proteins have different structures. You have the primary, secondary, tertiary, and quaternary. I want you to refer to the primary structure. Primary structure is basically defined as the series of amino acids linked together by peptide bonds. If this series of amino acids will start folding, that's the time you get to form the secondary, tertiary, and quaternary structures. Of all the bonds involved in the structures of proteins, it is only the peptide bond that is considered as covalent bond. Covalent bonds are strong bonds, meaning they are not easily destroyed, while the bonds that form the secondary, tertiary, and quaternary structures of proteins are basically weak bonds. That's why if proteins, particularly enzymes, are exposed to factors that can denature them, they will most likely lose their quaternary structure, tertiary, and secondary structures. And what will only be left is the primary because it is held together by peptide bonds. Shown in the picture is a tertiary protein, which is a good representative of an enzyme. If this protein or enzyme will be subjected to denaturation, this protein or enzyme will lose the folds within its structure and it will basically go back to its primary structure, which is composed of amino acids linked together by peptide bonds. At this point, we will be discussing how to name enzymes. There are several ways on how to name enzymes. We will just focus on how to name enzymes based on enzyme commission nomenclature. They usually give enzymes assigned numbers. And these assigned numbers are usually composed of 4 groups of numbers. We have the first digit, the second digit, the third digit, and the fourth digit. In naming enzymes based on enzyme commission nomenclature, we usually start writing the letters E and C. And this E and C denote enzyme commission. Then the first number that we should write down should reflect the class of the enzyme. And the class of the enzyme will be followed by another number, and that will represent the subclass of that particular enzyme. The subclass will be followed by a subclass of that enzyme, and the fourth and the last digit will be representing the serial number of the enzyme. Now, of all the numbers there, the most important for our lecture is the first digit, the class of the enzyme. Enzymes are classified based on the type of reactions they catalyzed. And we have here numbers 1 to 6. So, every time that you will see that, let's say for example, there's an enzyme which is named EC2.3.4.5, the number 2 in there represents the class of the enzyme. And that is now the type of reaction that the enzyme is catalyzing. So the number 2 in the name of the enzyme would mean that the enzyme is a transferase enzyme. So what are now the types of chemical reactions that the enzymes would catalyze? We have number 1, oxidoreductase. So any enzyme with the name EC followed by the number 1 would mean that the enzyme is an oxidoreductase. 2, transferase. 3, hydrolase. 4, lyase. 5, isomerase. 6, ligase. Lil. So before you move to the next part of the discussion, make it a point that you should memorize the specific number and the type of class the enzyme would belong to. Auth Lil. An oxidoreductase catalyzes oxidation reduction process. What do we mean by oxidation and reduction process? This is basically the transfer of hydrogen and oxygen, or simply electrons among the substrates to form the products. So take a look at these substrates A and B, and then we have here the products A and B. What do you notice? The electrons in A were transferred to B. So in the process, substrate A lost electrons. And when a substrate will lose an electron, it will become oxidized. If the substrate will gain electron, it will become reduced. So in the chemical reactions, A became oxidized, B became reduced. And since this reaction involves the transfer of electron, the enzyme that can catalyze this reaction is an oxidoreductase. Let me give you an example of a chemical reaction catalyzed by an oxidoreductase. This chemical reaction is catalyzed by lactate dehydrogenase. So take a look at the substrate first. You have the pyruvate and you have NADH. What do you notice? After the chemical reaction is catalyzed by lactate dehydrogenase, you can see that pyruvate gained an electron in the form of hydrogen and it became lactate, while the NADH lost electron and became the NAD+. So in this chemical reaction, pyruvate was reduced to form lactate and NADH was oxidized to form NAD+. Why oxidized? Because it lost an electron in the form of hydrogen. I know some of you are wondering, hydrogen is positively charged, H1+. How come the transfer of hydrogen will cause the chemical to become reduced? Please take a look at the picture of the hydrogen. For one proton of hydrogen, there's also one electron. So if you gained hydrogen, you gain its electron. Therefore, you are reduced. This is another example of a chemical reaction that is catalyzed by an oxidoreductase enzyme. So you have here the substrates malate and NAD+. And what do you notice? With the help of malate dehydrogenase, malate became oxaloacetate and NAD+ became NADH. So where did that hydrogen in NADH came from? It came from malate. So malate lost an hydrogen and became oxaloacetate. So if malate lost an hydrogen, it has lost an electron, therefore it is oxidized. While NAD+ gained hydrogen, with hydrogen it gains an electron, so therefore NAD+ was reduced to form NADH. The reason why the enzyme is named malate dehydrogenase because it catalyzed the removal of hydrogen from malate and transferred the hydrogen to NAD+. A transferase enzyme catalyzes the transfer of a functional group from one substrate to the other. In the case of oxidoreductase, we are just simply transferring hydrogen or oxygen or an electron. In the case of a transferase, we're transferring now a functional group. A functional group can be a methyl group, an acyl, an amino group, or a phosphate group. So take a look at this chemical reaction. You have here alanine, and alanine is made to react with oxaloacetate to form the products pyruvate and aspartate. What do we notice? The amino group of alanine was transferred to oxaloacetate. That's why the oxaloacetate gained the amino group and it became the aspartate. And alanine, after it has lost its amino group, became the pyruvate. So there is the transfer of a functional group in this chemical reaction. So the enzyme involved is most likely a transferase enzyme. Let's have another example. This chemical reaction is catalyzed by aspartate aminotransferase. So let's dissect the name of the enzyme. Aspartate aminotransferase. So this enzyme will transfer an amino group. Most likely the amino group will come from the substrate aspartate. So we have here the substrates aspartate and ketoglutarate, and the products glutamate and oxaloacetate. We have here the amino group of aspartate, and that amino group of aspartate will be transferred to ketoglutarate. Ketoglutarate will become the glutamate after it has gained the amino group, and aspartate became the oxaloacetate after losing the amino group. And since what is transferred here is an amino group, it's a functional group, then this enzyme, aspartate aminotransferase, is a transferase enzyme. And if you're going to give it a name under enzyme commission, you will write down the name as EC2 because the first number would always reflect the class of the enzyme. And this enzyme is a transferase, and transferases enzyme are assigned with number 2. Let's have another enzyme, hexakinase. The substrates are glucose and ATP. And then what do you notice? One of the phosphate group of ATP was incorporated in the chemical structure of glucose, converting it into glucose 6-phosphate. The number 6 there would indicate that the phosphate group was made to attach on the 6th carbon of glucose. And ATP became ADP. So there's a transfer of a phosphate group. And phosphate group is a functional group. So therefore, hexakinase is a transferase enzyme. Kinase would always mean that what is being transferred is a phosphate group. And the word hexo there would refer to glucose because glucose has 6 carbons. Hexakinase will transfer phosphate group involving a sugar with 6 carbons. The third class of enzyme is hydrolase. Hydrolases catalyze chemical reactions that form two products from a large substrate by breaking down bonds with addition of water. So, the breakdown of bonds should involve the addition of water. So, take a look at these two monosaccharides linked together by a glycosidic bond. Upon the addition of water, the glycosidic bond was broken, and you have here two separate monosaccharides. One large substrate being converted to two small products. A good example of a hydrolase enzyme is lipase. Lipase is used by the body to break down triglyceride. So I want you to take a look at the triglyceride in the chemical reaction. It is composed of a glycerol backbone, and attached to the glycerol backbone are 3 fatty acids. Now, what will happen to this triglyceride if it will be acted upon by the lipase? The lipase enzyme will act on the ester bonds. These ester bonds will be made to react with water as shown in the picture. And what will happen? With the addition of water, these ester bonds will be broken down, and the 3 fatty acids will now be separated from the lipase enzyme. How? The hydrogen from water will bind to the oxygen of the glycerol, while the OH of water or hydroxyl group of water will combine or react to the fatty acids, separating now the fatty acids from the glycerol. So, what made this chemical reaction possible? What separated the fatty acids from the glycerol by breaking down the bonds is the addition of water. So, the enzyme that will catalyze this reaction is most likely a hydrolase enzyme. The fourth type of enzyme is similar to hydrolase. It will catalyze the breakdown of bonds in a substrate. But this time, the breakdown of bonds will not involve the addition of water. And this enzyme is referred to as lyase. A good example of a lyase enzyme is argininosuccinate lyase. As you can see in this picture, a certain bond in argininosuccinate will be broken down by the enzyme. forming now arginine and fumarate. But the breakdown of the chemical bond did not involve hydrolysis or addition of water. The next enzyme is isomerase. Isomerases catalyzed reactions that involved intramolecular arrangement of functional groups or molecules within the substrate. So for you to understand that, take a look at the example chemical reaction. You have here 3-phosphoglycerate to be acted upon by phosphoglycerate mutase. Take a look at the phosphate group of phosphoglycerate. This phosphate group is attached to its third carbon. But with the action of phosphoglycerate mutase, that phosphate will be transferred to its second carbon. There is a rearrangement of the order of molecules and functional group within the substrate in the chemical reactions catalyzed by an isomerase. And since in the example, the phosphate group did not come from another substrate, it came from the same substrate, it's just that it was transferred from one carbon to the other. So this is an example of a chemical reaction catalyzed by an isomerase enzyme. For you to understand the activity of an isomerase enzyme, I'll give you another example. This time, you have the enzyme phosphohexose isomerase. Phosphohexose, meaning it's going to act on a sugar that has 6 carbons and there's a phosphate attached to one of the carbons. And that substrate is glucose 6-phosphate. Glucose is a 6-carbon containing carbohydrate and it has a phosphate group attached on its 6th carbon. Now, what will this enzyme do? It will just simply rearrange the carbons, the hydrogen, and the oxygen atoms within the structure of glucose, converting it into fructose 6-phosphate. As you can see, none of the carbon atoms, hydrogen, and oxygen atoms were removed. It's just that they were just rearranged. And this is basically an activity of an isomerase enzyme. The last but not the least, the ligase enzyme. If hydrolase and lyase would break bonds to produce the products, in the case of ligase, it will actually join two substrates, form a bond between them to form a product. A good example of a ligase enzyme is DNA ligase. It will basically join two DNA strands together. Let's have a review on how to name enzymes based on the Enzyme Commission nomenclature. We usually write first the letters E and C, and these letters will be followed by the first digit. The first digit would always reflect the class of the enzyme, the auth lil that you memorized a while ago. The first digit will be followed by the second digit, which would reflect the subclass. The third digit will reflect the sub-subclass, and the fourth digit will reflect the serial number of that particular enzyme. So you have in this photo the list of the classes of the enzyme. You have their numbers 1, 2, 3, 4, 5, 6. Now, I will just show you how to come up with the subclass and the subclasses of these enzymes. So I will use ligase as an example. Now, under the group of ligases enzyme, you have there classes of enzymes that form bonds between two substrates to form a product, and the bonds would involve the union of carbon and oxygen. You also have a type of ligase enzyme that will join two substrates together to form a product by forming a bond between carbon and sulfur. You also have another type of ligase enzyme, a subclass that would join two substrates together to form a product by forming a bond between carbon and nitrogen. So as you can see, you first group the enzyme according to the class. You know that they form products by joining two substrates together, so you will all refer to them as ligase. Then after coming up with the class, you go with the subclass. In the level of the subclass, you have to identify what bonds are formed by the particular enzyme. So you will give the enzyme name EC6.1 if the bond formed by that particular ligase enzyme would involve the union of or the bond formation between carbon and oxygen. You will give it the name EC6.2 if it will particularly form a bond between carbon and sulfur. So this 6.1, 0.2, 0.3 are referring to the subclass of the enzymes that belong to the ligase. Now, what we will do is, let's identify the subclasses. So let's focus on the EC6.3. 6.3 is already revealing to us the subclass of the enzyme. Let's go with the subclasses. Under the enzymes that can form bonds between carbon and nitrogen, we also have to identify if the carbon and nitrogen bond is formed between an acid and ammonia. So we will give that enzyme the name EC6.3.1. But if the carbon and nitrogen that were joined together is between an acid and an amino acid, then we will give that enzyme EC6.3.2. The two there would reflect now the subclass of that particular enzyme. So I hope at this point, you now know the meaning of the class, the subclass, and the subclasses of the enzyme. Let's have definitions of some terms. We have the term substrate and product. The substances acted upon by the enzyme will be referred to as the substrates. And please take note, enzymes are always specific to their substrates, while the chemicals that would result from the enzymatic chemical reaction will now be referred to as the products. The part in the enzyme where the substrate will bind into and the area where the substrate will undergo the chemical reaction is called the active site. So please take a look at the enzyme in the picture. It's colored blue. You have here the portion that is labeled as active site. It is in that active site that the substrate will bind into the enzyme. Sites other than the active site, so basically the other sites in the enzyme, will now be called as the allosteric site. In terms of the active site, it has two important portions. You have the binding site and you have the catalytic site. The binding site, as shown in the picture, is the portion of the active site that will attach itself or bind to the substrate. And the catalytic site is the site in the active enzyme that will now perform the chemical reaction. Do you know that enzymes would require other chemicals to be present first before this enzyme will become functional or active? Enzymes alone, you call them as apoenzyme, they are inactive. But with the presence of a cofactor, a chemical that will activate them, this apoenzyme will become the holoenzyme. The holoenzyme now is referred to as the active enzyme. So without the cofactors, this enzyme will not be able to catalyze the chemical reactions. So apoenzyme is the protein part of the enzyme. It's basically the inactive form of the enzyme. And this apoenzyme would need the presence of a cofactor, and the presence of the cofactor will convert the apoenzyme into the active holoenzyme. So it is the holoenzyme that is catalyzing the chemical reactions. Enzymes are proteins, and the non-protein chemicals that they need to convert them from apoenzyme to holoenzyme are called cofactors. Without these cofactors, the enzyme will remain inactive or as an apoenzyme. Cofactors can be inorganic or organic. Inorganic cofactors are most of the time metals, such as zinc, iron, and copper. When you say organic cofactors, these chemicals should contain carbon, hydrogen, oxygen, and sometimes nitrogen in their chemical structure, just like the other organic compounds that we know. So take a look at the chemical structure of one of the cofactors. Can you appreciate that it has carbon, hydrogen, oxygen, and nitrogen? Now, take note, organic cofactors are further classified as prosthetic, and coenzyme. One of the popular inorganic cofactor is magnesium. And magnesium is the cofactor of the enzyme hexakinase. And how important is hexakinase? It's the enzyme that is involved in the first chemical reaction in glycolysis. It is involved in the transfer of phosphate group from ATP to glucose, converting the glucose now to glucose 6-phosphate. But the thing is, I'm just using this one as an example to highlight that this enzyme would always require an inorganic cofactor in the form of magnesium. This chemical reaction is catalyzed by phenylalanine hydroxylase. And phenylalanine hydroxylase would need an organic cofactor for it to become functional. And the organic cofactor I am referring to is biopterin. This is the chemical structure of biopterin. So it's very obvious it's an organic compound. Now, what happens in the chemical reaction is that the phenylalanine is acted upon by phenylalanine hydroxylase with the help of biopterin, and phenylalanine is converted to tyrosine. Tyrosine will now be used by the body as a substrate for the production of melanin. There are people born with phenylketonuria, meaning they don't have the genes that encode for the enzyme phenylalanine hydroxylase. So expect that these people will not be able to convert phenylalanine to tyrosine. Therefore, they won't have a substrate for the production of melanin. That's why patients with phenylketonuria will present with albinism. The problem with phenylketonuria, there will be a buildup of phenylalanine in the blood. The excess phenylalanine will be converted to phenylactate and phenylacetate, which are known to be neurotoxic. That's why children born with phenylketonuria will most likely develop mental retardation if not diagnosed immediately after birth. Do you know that around 30% of phenylketonuria cases in Southeast Asian children is not because they don't have the gene that encodes for the phenylalanine hydroxylase enzyme? The phenylketonuria among these children is brought about by the deficiency of the biopterin. And that will only prove to you guys that the absence of cofactor will convert the enzyme from holoenzyme to apoenzyme. And apoenzyme is the inactive form of the enzyme. Therefore, this phenylalanine hydroxylase cannot convert phenylalanine to tyrosine because of the absence of its cofactor. Organic cofactors can be further classified as prosthetic or coenzyme. When you say coenzyme, this organic cofactor will bind loosely to the enzyme. So meaning, if you have the enzyme, this coenzyme will just bind to the enzyme during the chemical reaction. After the chemical reaction, that coenzyme might detach from the enzyme. But if the organic cofactor is tightly bound to the enzyme, then you will refer to it as a prosthetic group. Let's have a summary. Enzymes need cofactors for them to be converted from apoenzyme to holoenzyme. And these cofactors are considered as the non-protein portion of the enzyme because it is the apoenzyme that is the protein portion. Cofactors can be divided into inorganic or organic cofactors. Inorganic cofactors are most of the time metals such as magnesium, calcium, zinc, copper, okay? And we have the organic cofactors. And organic cofactors are further divided into prosthetic group and coenzyme. If the organic cofactor will detach from the enzyme after the catalysis of the chemical reaction, then it's considered as a coenzyme. If it is tightly bound to the enzyme, then you will refer to it as prosthetic group. Again, the apoenzyme is the protein portion of the enzyme, while the non-protein portion is called the cofactor. Combined together, they will form the holoenzyme. At this point, we will be discussing enzyme kinetics. For you to understand this topic, you have to bear in mind this chemical reaction. You have here the substrate and the enzyme. And then later on, they will form the enzyme substrate complex. And after that, the product is formed and the product is released from the enzyme to regenerate the free enzyme so that that free enzyme can act on another substrate and form new products. For us to understand enzyme kinetics, it's very important to know the Michaelis-Menten graph. Plotted on the y-axis of the graph is the rate of reaction. Plotted on the x-axis of the graph is the substrate concentration. According to Michaelis and Menten, if you're going to plot on a graph the velocity of an enzymatic reaction, you won't see a straight line. Instead, you will see an S-shaped curve. That's why they describe the curve to be hyperbolic. According to Michaelis-Menten kinetics or graph, at low concentration of substrate, the rate of the enzymatic reaction will increase as you increase the substrate concentration, and it's very obvious in the graph. As the concentration of the substrate increases, you can also see a subsequent increase in the rate of reaction as shown on the y-axis. Why do you think there is an increasing rate of reaction? Because there are still enzymes with unoccupied active sites. And once you add substrate to the chemical reaction, then these enzymes will act on the substrate. Thereby, they can increase the rate of reaction. So, you call this one as first-order reaction. In first-order reaction, as you increase the substrate concentration, the rate of enzymatic reaction will also increase. So, the rate of reaction is dependent on the substrate concentration. Again, at first-order reaction, as you increase the substrate concentration, the rate of reaction will also increase. Because there are still available enzymes, and once substrate is made available to them, they will immediately act on the substrate, resulting to the increase in the rate of chemical reaction. So in first-order reaction, the rate of reaction is dependent on the substrate concentration. But there will come a time that all of the enzymes will be saturated with the substrate, meaning there are no more free enzymes that can act on the substrate that you're adding into the chemical reaction. Most of you would say, I expect that the chemical reaction rate should go down because there are no more reactions happening. Please take note, even if the enzymes are saturated, enzymes are not consumed during the chemical reaction. So as soon as one enzyme is done converting the substrate into the product, that enzyme will accommodate another substrate. That's why, as you can see on the graph, the chemical reaction will reach a plateau. Plateau, meaning it will not decline, it will just remain at a certain level. Why? Because the enzymes are not consumed. They will still continue the chemical reaction. But you won't see further increase because the enzymes are already saturated. And you call this one as zero-order reaction. In zero-order reaction, as you increase the substrate concentration, you won't observe any further increase in the rate of reaction. So what you need to memorize about the zero-order concentration is what you should change is not the substrate concentration. What you should change is the enzyme concentration. So if you want the chemical reaction at zero-order reaction to increase, you add more enzyme. So first-order reaction is dependent on substrate concentration in terms of increasing the rate of reaction. The zero-order reaction is dependent on the enzyme concentration. Let's have a review. In first-order reaction, you have there the increase in the rate of reaction as you increase the substrate concentration. Because enzymes are not yet saturated, you still have unoccupied active sites which can act on the substrates. But as soon as the rate of reaction will reach a plateau, you are already in the zero-order reaction, wherein all the enzymes are already saturated with substrates. The reaction will not decline because the enzymes are not lost during the chemical reaction. Once the enzyme is freed upon the production of the products, the enzyme will now act on the substrates that you added into the chemical reaction, maintaining the reaction in a plateau or steady state. The maximum velocity that the enzymatic reaction can reach is called the Vmax or the maximum velocity. Vmax represents the maximum velocity that an enzymatic reaction can reach. This is the velocity wherein the enzymes are already saturated with the substrate. So that is your Vmax. You also have to identify half of the Vmax, meaning half of the velocity that the enzymatic reaction can reach. So this is most likely 1/2 Vmax of our enzymatic reaction. The concentration of the substrate that matches half of the Vmax will be referred to as Km. So Km is the concentration of the substrate that is enough for the enzyme to reach half of its Vmax. So looking at this graph, you have here the Vmax. This is half of the Vmax, and the concentration of the substrate that is enough for the enzyme to reach the half of the Vmax is called as the Km. I hope you understand when I say that the rate of chemical reaction does not only rely on the substrate concentration. The substrate and the enzyme must have affinities to each other, because if they don't have affinities to each other, then the enzymatic reaction will not proceed. So, the affinity of the substrate and enzyme can be determined using the Km. Km, by the way, is the Michaelis constant. What do you think is the important? Therefore, we can conclude that the higher the Km, the lower the affinity of the substrate to the enzyme. And it would only mean that it would take a higher concentration of the substrate before the enzyme would be able to reach half of its Vmax. The lower the Km, the higher the affinity. You only need small amount of substrate for the enzyme to reach half of its Vmax. So what is now the relationship between Michaelis constant or Km and the affinity of the substrate to the enzyme? Inverse relationship. Because the higher the Km, the lower the affinity. What are now the factors that can affect enzymatic reactions? Number one, substrate concentration. Because you know very well, in first order concentration, as you increase the substrate concentration, the enzymatic reaction will increase. The further increase in the enzymatic reaction after the addition of substrate will no longer happen in zero order reaction. Because in zero order reaction, what will now increase the rate of reaction is no longer the substrate concentration but the enzyme concentration. So that will also tell us that the concentration of the enzyme is also a factor that can affect the enzymatic reaction. Another factor that can affect the rate of enzymatic reaction is the pH. pH can denature enzymes. Thereby, they can cause the enzyme to lose its activity. And we will discuss how the pH can denature enzymes later on. It's very important to take note that majority of the enzymes in the body would work best at a pH of 7.0 to 8.0. But there are also enzymes in the body that work best at an acidic pH or at an alkaline pH. A good example would be the enzyme pepsin. It digests proteins in the stomach, and it works best if it is mixed with hydrochloric acid because it is more active in an acidic pH. Another example will be alkaline phosphatase. The reason why it's called alkaline phosphatase because it works best at an alkaline pH. Enzymes are sensitive to pH just like the other proteins because changing the pH on its surrounding will cause the enzyme to change its shape. Remember, enzymes are proteins, and enzymes could have tertiary structures. Tertiary structures are formed by different bonds. One of the bond involved there is ionic bond. An ionic bond is formed between positively charged amino acids such as lysine, arginine, and histidine. They are positively charged because they have extra amino group in their R groups. Positively charged amino acids and negatively charged amino acids such as aspartate and glutamate. They are negatively charged because they have extra carbolic acid in their R side chain. Now, if the protein has both positively charged amino acids and negatively charged amino acids in its structure, these amino acids will form now what we call the ionic bonds. Ionic bonds are one of the bonds that form the tertiary structures of proteins. And as mentioned a while ago, the bonds involved in secondary, tertiary, and quaternary structural proteins are weak bonds. So they can be subjected to destruction, causing the protein to undergo denaturation. How is this one related to pH? Let's say, for example, this tertiary protein or enzyme is placed in an acidic pH. And when you say acidic pH, there is the abundance of hydrogen. Hydrogen is positively charged. And what will happen is the negatively charged amino acid might no longer bind or interact with the positively charged amino acid to form the ionic bond. The negatively charged amino acid will now interact with hydrogen. So, the tertiary structure has already lost one of its ionic bonds. Therefore, this enzyme or tertiary protein will become denatured. Same is true with placing the enzyme in an alkaline pH because the hydroxyl group or the OH negative of the alkaline solution will interact with the positively charged amino acid, causing it not to interact anymore with negatively charged amino acid and the loss of ionic bond. Another factor that can affect the rate of enzymatic reaction is temperature. If you're going to increase the temperature, you are making the molecules move. And when they move, they collide with each other, and then these molecules can form the products. So therefore, increasing the temperature can hasten chemical reactions, particularly the chemical reactions catalyzed by the enzyme. And according to some references, for every 10-degree increase in that temperature, the reaction rate is actually doubled. However, as you increase the temperature, you are destroying the weak bonds that form the quaternary, tertiary, and secondary structures of the proteins and enzymes, causing them to become denatured. So take a look at the graph. Plotted on the x-axis is the temperature, plotted on the y-axis is the rate of reaction. As you increase the temperature, you can expect to find or to see increasing rate of chemical reaction. But there will come a time, that the temperature will now cause the protein to become denatured and you will see a drop in the rate of enzymatic reaction. According to one of the references I've read, denaturation of enzymes would occur starting from 40 to 50 degrees Celsius. The temperature at which the maximum rate of enzymatic reaction occurs is called the optimum temperature. And take note, different enzymes have different optimum temperature, but majority of the enzymes that we have in our body have optimum temperature of 37 degrees Celsius, and that is our body temperature. Cofactors can also affect enzymatic reaction because their absence would convert the holoenzyme to apoenzyme, and that's an inactive form of the enzyme. So what do you think will happen? The absence of cofactors will slow down the rate of enzymatic reaction. Inhibitors can also affect the rate of enzymatic reactions, and we have three types of inhibitors, and they have different mechanisms of actions on how to inhibit enzymatic reactions. We have competitive, we have non-competitive, and uncompetitive type of inhibitors. Competitive inhibitors resemble or look like that of the substrate. Therefore, they can bind to the active sites of the enzymes. If they do, they will now hinder the binding of the substrate to the active site of the enzyme, and that will have an effect on the rate of the enzymatic reaction. What is good about competitive inhibitors is that the effect of this type of inhibitors is reversible. And how do you think we can reverse the effect of a competitive inhibitor? For you to understand how to reverse the effect of a competitive inhibitor, I want you to take a look at these actors of the series The World of the Married. The guy would be our enzyme, the wife would be our substrate, and the heart of the guy is the active site. If this guy will meet a chemical that resembles his wife, then most likely that chemical can also bind to his heart. So this chemical now is acting as the competitive inhibitor. How to reverse the effect of this type of competitive inhibitor? You just have to increase the substrate concentration. You have to remind the guy who is the real boss. Increasing the concentration of the original substrate will cause the displacement of the competitive inhibitor. What do you think now will happen to the enzymatic reaction in the presence of a competitive inhibitor? The purple line is representing the enzymatic reaction without the competitive inhibitor. The yellow line is representing the enzymatic reaction in the presence of the competitive inhibitor. What do you notice? The Vmax was still reached by the yellow line given that there is a competitive inhibitor, but it only reached the Vmax at a higher substrate concentration compared to the purple line. Why do you think it's possible? Because the effect of competitive inhibitor is reversed if you increase the substrate concentration. That's why in these two types of reaction, without the competitor, without the competitive inhibitor, and with the competitive inhibitor, the Vmax will still be the same. So if the Vmax are the same for the two types of reactions, expect that the half Vmax will also be the same. The thing now is, what will be the change in the Km, if there is any? Let us figure out what will happen to the Michaelis constant or Km in the presence of competitive inhibitor. This is the Vmax for both reactions. This is half of the Vmax for the reaction without the competitive inhibitor. And this is its corresponding substrate concentration or the Michaelis constant or Km. This is half of the Vmax for the enzymatic reaction with the presence of competitive inhibitor. It's basically the same as that of the reaction without the competitive inhibitor. And this is its corresponding substrate concentration or Km. Is there any change? Yes, the Km of the enzymatic reaction with the competitive inhibitor is higher than that of the enzymatic reaction without the competitive inhibitor. And Km reflects the affinity of the substrate to the enzyme. So meaning, in the enzymatic reaction with the competitive inhibitor, the affinity of the substrate is lower because the higher the Km or Michaelis constant, the lower the affinity of the substrate to the enzyme. And it is expected because the enzyme will be torn between the substrate and the competitive inhibitor. So therefore, the enzyme will have lesser affinity to the substrate if there is the presence of a competitive inhibitor. Thereby, the Km will really increase. But both the Vmax and half the Vmax will be the same. A non-competitive inhibitor at the same time will not compete with the substrate at the active site. Instead, it will bind to an allosteric site, and the binding of this non-competitive inhibitor to the allosteric site will cause a change in the shape of the active site. So take a look at letter B in the photo. You have there the yellow-colored non-competitive inhibitor. It did not compete with the substrate at the active site. It is bound to the allosteric site of enzyme B in the picture, and it caused the active site of enzyme B to change. Therefore, the active site cannot anymore accommodate the substrate. So expect the enzymatic reaction will slow down. Letter A is showing us a competitive inhibitor. The blue figure there is a competitive inhibitor, and it is binding at the active site, hindering now the binding of the substrate to the enzyme. But this one is reversible. You can just simply displace the competitive inhibitor by increasing the substrate concentration. In the case of letter B, a non-competitive inhibitor, the non-competitive inhibitor is bound to the allosteric site of the enzyme, causing now a change in the shape of the enzyme and as well as its active site. Therefore, the active site cannot accommodate the substrate and begin the chemical reaction. The problem with non-competitive inhibitors is that their effect is irreversible, meaning you cannot anymore correct the rate of enzymatic reaction by increasing the substrate concentration. This is how the non-competitive inhibitor will inhibit the binding of the substrate to the enzyme, the wife to the heart of the husband. The non-competitive inhibitor knows that the best way to a man's heart is through his stomach. So this non-competitive inhibitor will not go to the active site, which is the heart. It will go to the allosteric site, which is the stomach. And giving the guy biscuit will cause a change of heart. And the guy's heart now, or active site, will not react to the wife substrate. In the case of non-competitive inhibitor, the wife will become sad and the non-competitive inhibitor will win. Now, what will happen to the enzymatic reaction given that you have the presence of a non-competitive inhibitor? Purple line is representing the enzymatic reaction without the presence of the non-competitive inhibitor. Green line is representing the enzymatic reaction with the presence of non-competitive inhibitor. What do you notice? There's a significant drop in the Vmax. You cannot anymore reach the former Vmax because the reaction is irreversible. So if the Vmax is significantly reduced, expect that the half Vmax will also be reduced. But what do you think will be the effect on the Michaelis constant or Km? This is the Vmax of the enzymatic reaction without the presence of the non-competitive inhibitor. This is the Vmax of the enzymatic reaction in the presence of the non-competitive inhibitor. It's very obvious that the Vmax significantly reduced. This is half of the Vmax of the chemical reaction without the presence of the non-competitive inhibitor, and this is its corresponding Michaelis constant. This is the Vmax of the enzymatic reaction in the presence of non-competitive inhibitor. So as you can see, it also significantly decreased, similar to the Vmax. And this is its corresponding Michaelis constant. In the case of non-competitive inhibitor, the Km remains the same. Why do you think so? Why do you think the Km remains unaffected in the presence of non-competitive inhibitor. So I'll give you 10 seconds to think. In the presence of non-competitive inhibitor, as long as the enzyme is not yet affected by the inhibitor, the enzyme will bind to the substrate and retain the same amount of affinity to the substrate. Because anyways, non-competitive inhibitor will not bind to the active site. They will bind to the allosteric site. So if you have remaining unaffected enzymes, they will bind directly to the substrate with the same amount of affinity. That's why the Michaelis constant and the Km will remain the same. This is not true with competitive inhibitors because in competitive inhibitors, you have two chemicals binding to the same site, which is the active site. So the enzyme will be torn between the substrate and the competitive inhibitor and expect that the Km will become increased and the affinity of the enzyme to the substrate will become lower. But in the case of non-competitive inhibitor, as long as the enzyme is unaffected by the non-competitive inhibitor, it will bind directly to the substrate. That's why you can still see that the rate of reaction is still there. Okay? So, same amount of affinity to the substrate as long as the enzyme is not bound to the non-competitive inhibitor. But then again, even if you will increase the substrate concentration, you will never ever reach the former Vmax. The third type of inhibitor is the uncompetitive inhibitor. This type of inhibitor does not bind to the enzyme alone. It will wait for the enzyme to bind to the substrate and it will now bind to the enzyme substrate complex and not allowing the two to dissociate to form the products. And take note, uncompetitive inhibitors have irreversible effects as well, similar to non-competitive inhibitors. Uncompetitive inhibition can be seen in the stories of some Filipino teleseries wherein the third party or the mistress will hire goons to kidnap the main lovers, and then the mistress or the third party will try to kill them so that they will not have an happy ending. So the mistress or the third party is the uncompetitive inhibitor. The main characters or the lovers are the enzyme substrate complex. So the uncompetitive inhibitor will not allow the enzyme substrate or the main lovers to have a happy ending or for the enzyme substrate to end up with producing the products. Again, uncompetitive inhibition takes place when an enzyme inhibitor binds to the enzyme substrate complex. What will now happen to the enzymatic reaction in the presence of an uncompetitive inhibitor? Blue line is representing the enzymatic reaction without the inhibitor. Red line is representing the enzymatic reaction in the presence of uncompetitive inhibitor. What do you notice? The Vmax is significantly reduced. So expect that half Vmax will also be reduced. You will be surprised to find out that the Km or Michaelis constant of the reaction with the uncompetitive inhibitor is lower than the enzymatic reaction with no inhibitor. So this would signify that the substrate in the presence of uncompetitive inhibitor would have a higher affinity to the enzyme. And what do you think is the reason behind it? So I want you to think for the reason. I'll give you 10 seconds. Remember, the uncompetitive inhibitor will bind to the enzyme substrate complex and it will not allow the two to dissociate from each other to produce the products. So if the two are not allowed to dissociate from each other, they would have a higher affinity to each other, and that will now be seen by the lower Michaelis constant value or lower Km value. So There are two models on how substrate will bind to the enzyme. The first that was proposed is Fisher's lock and key model. This model assumes that the active site of the enzyme and the substrate have the same shape, just like the key and the lock. This model supposes that the substrate fits perfectly to the active site of the enzyme. The lock and key model was later replaced by the induced fit model by Koch-Lantz. This model presumed that the enzyme molecule does not retain its original shape after it will bind to the substrate, meaning the active site does not have the same shape as that of the substrate. And after the substrate will bind to the enzyme, the active site will have a configurational change. It will try to change its shape to accommodate now the substrate. So this model emphasizes that proteins and enzymes are flexible. That's why they can adjust the shape of their active sites just to accommodate the substrate. The lock and key model portrays an enzyme as rigid structure, that the active site must have the same shape as that of the substrate. The induced fit model, on the other hand, is portraying the enzyme as a flexible structure, that it will allow its active site to have a conformational change or a change in the shape to accommodate the substrate. Now, supporters of lock and key would say the lock and key would always emphasize the specificity of the enzyme to the substrate, because the enzyme will only act to the substrate with the same shape as the active site of the enzyme. So thus, induced fit model suggests that enzymes can basically act on any substrate as long as they will change their active site to accommodate the substrate. The answer of the supporters of induced fit model to that question is that the substrate must do more than just simply fit into the active site. Now, please recall that the active site has two portions. You have the binding site and you have the catalytic site. Induced fit model would say that on the active site that has not yet attached to the substrate, the catalytic site is not yet formed. And the catalytic site will only be formed after the substrate will bind to the active site and cause it to change its shape. That's why the blue arrow now is showing you the catalytic site of the active site, which was not formally present in the active site that has not yet bound to the substrate. So therefore, today, induced fit model is the acceptable model for the interaction of substrate to the enzyme. This is Dr. Salaver, and I would like to thank you for listening to our first lecture on enzymes. Going down the Michaelis constant or Km of enzymatic reactions. So take a look at the graph. You have there three chemical reactions. Do not mind the yellow line. That is representing a chemical reaction that is not catalyzed by an enzyme. The two other lines, the blue and the red, are representing enzymatic reactions. Please assume that they have the same Vmax. So expect that they would have the same half Vmax. I want you to identify the Km or Michaelis constants of these two enzymatic reactions. Remember, that's the concentration of the substrate at half of the Vmax or of the enzymatic reaction. Now, this is the Michaelis constant or Km of enzymatic reaction 1, the blue line. And this is the Michaelis constant or Km of enzymatic number 2, red line. What do you notice? Blue line or enzymatic reaction 1 was able to reach the half of its Vmax at a lower substrate concentration. while the red line or the enzymatic reaction number 2 needed a higher substrate concentration before it was able to reach half of its Vmax. So therefore, Km or Michaelis constant can show us the affinity of the substrate to the enzyme. That the substrate, even at low concentration, has already bound to the enzyme and was acted by the enzyme immediately. That's why both of them was able to reach half of the Vmax at a low concentration of the substrate.